particular one of the particular focuses is to really just help underscore the power of science, to be able to address some of the more significant um, public health crises that we've had. I'm going to focus on two: HIV, which I've spent over 35 years of my life focused on, and then obviously as CDC director, and since then, the COVID-19 pandemic, and use them as examples. While I'm a big advocate and have been about the importance of heavy investment, particularly in NIH and the discovery dollars that are coming in, I'm also a big advocate that there needs to be funding for the application of discovery when discovery occurs to impact clinical medicine. And unfortunately, I think our nation is pretty out of balance with that. There's a lot of discovery that we develop that really takes a great deal of time to actually penetrate and have an impact, public health impact, clinical impact on the human condition. I think one of the great examples I used to use to try to illustrate this to Congress was hepatitis C. As a clinical virologist, it wasn't long ago when many of us thought this was a very difficult therapeutic challenge. And lo and behold, the power of science had not only reduced hepatitis C to a treatable disease, but it actually reduced it to a curable disease. My first year as CDC director, 18,000 people died of hepatitis C. And I want to note that that was 5 years after curative therapy was available. I want to congratulate the VA, who I think is one of the more forward moving groups in making sure the therapeutic advancements that science is given to hepatitis C get applied to patients, but many parts of our nation continue that's a great scientific advancement, but it has no practical implication in the lives of literally tens of thousands of people living with hepatitis C. So that's part of one of the themes that I want to talk about: power of science. But if that science doesn't get applied to impact public health, one has to really wonder what's the driving force behind that science. I started with a reflection that I used mostly through my career, which for those of you who aren't counting now, I just turned 70. So it's you're getting up there. My wife also turned 70. She's not a happy camper when I remind her we're in our 8th decade of life. She thinks it makes us sound older than we really are. But the fact is we're in our 8th decade of life. And this quote, to me, always meant a great deal about preventive medicine. Preventive medicine dreams of a time when there be no unnecessary suffering, no premature death, when the welfare of people shall be the highest concern, when humanity and mercy shall replace greed and selfishness. And it dreams that all of these are accomplished through the wisdom of man. And really, that's another way of saying the power of science. And those people who know me, I also, obviously have a strong faith, and I believe that science is one of the many gifts that God has given our world to be able to improve the human condition. I thought I'd start with HIV as an example and try to juxtapose it a little to the COVID pandemic that we have right now. Hard to believe when I was, 31 years old, 32 years old, 33 years old, I became attracted to this new occurrence that originally was reported in a handful of individuals in Los Angeles and New York, which went by a variety of different names, eventually AIDS, and began to get fully engaged initially in the care and treatment of young people that were not that younger than myself, with an illness that, despite what modern medicine and I tried, frequently ended in premature death. And I had an opportunity to get involved in trying to be parts of teams. I got to work with two really important groups. I got to work with, obviously, Bob Gallow's group at NIH, and I also got to work with Tony Fauci's group at NIH, as both of those teams were trying to unravel some of the mysteries of this new initially what people referred to as a medical curiosity that occurred in really a handful of individuals. And between 1981 and 1984, which is about 3 years, there was an enormous effort to apply public health epidemiology, clinical observation, and more importantly, basic scientific discovery to confront this new syndrome, AIDS, an aggregate of signs and symptoms without an etiology. And I think it's remarkable, and again, I use this term very carefully when I said I actually think it's miraculous, that in between 1981 and 1984, the power of science, one of your own faculty, Bob Gallo, was really the driver of much of this, reduced this mysterious syndrome to a disease that is a disorder of defined etiology. That caused by a new virus, a new human virus, a new human retrovirus, which eventually became known as HIV, fully articulated its means of transmission. Basically, it's a sexually transmitted disease. It was also transmitted by blood and blood products, and it could be transmitted by the intimacy of a mother to a newborn child, either through birth or breast milk. And in that period of time, again, about 3 years, science, again, the Gallup group really driving this, developed a diagnostic test. So no longer did clinical medicine have to wait to the end stages of AIDS to make this diagnosis. We now had a test that could allow us to define if someone was infected with HIV literally 5-10 years prior to developing significant clinical disease. And that test eventually, obviously, got applied in different circumstances, particularly in blood transfusions, and saved many different lives. Although there was a disconnect, and we'll come to that in a second, in terms of the ability of this discovery and the ability to have a new diagnostic test for it to be integrated into clinical medicine to make the public health impact that it eventually would make. We went through a very long period, 1986, I'm going to say, until about 2003. I know I was involved working with colleagues at NIH, Sam Broder in particular, in the first trial of an antiviral AZT. I think if I remember correctly, it was in somewhere between 15 and 20 patients. And I know that we were relatively excited because rather than a life expectancy under a year, we were able to have these individuals survive about 26 months. And so that was viewed as the sort of the beginning that the potential to develop antiviral therapeutics may be a goal that could be reached. Now, in retrospect, I think a lot of the therapeutic advancements of that early trial had more to do with pneumocystis prophylaxis than it did the initial antiretroviral therapy with AZT. But it also opened a time between 86 that a variety of different therapeutics were developed. And while the protease inhibitors made a big splash back in 96, because it actually was a combination that finally led to some people to get viral suppression to the point that they didn't advance in disease. It really wasn't until about 2003 when the therapeutic advancements were such that a highly active, globally available antiviral therapy was now available to treat HIV infection. Again, facilitated enormously by President Bush's decision to advance the PEPFAR program, which many of the people at Maryland played a very important role in, and really became, by 2003, you could effectively treat most individuals with HIV infection here and around the world with antiretrovirals, so that no longer when 1981, 82, 83, most of the men and women that I had the privilege to serve as their doctor basically had, less than a 12, 18-month survival. Now, all of a sudden, we had the therapeutic tools that can allow individuals, even with extremely advanced disease, when they presented, T cell counts really you couldn't measure them. I would just talk to one of my early patients on the phone shortly before this talk, and I remember her quite well with her dear friend. They were both my patients, and her friend, unfortunately, in 1995 died before we had access to the protease inhibitors, which helped stabilize many patients. But this other young lady, we were able to stabilize despite having no T cell count for more than a year. And obviously today she's a robust, highly effective grandmother, and lived an enormously productive life, serving many of the people trying to live with HIV successfully in Baltimore. So those therapeutic advancements were huge. But part of what I want to focus on about the reflections is that despite that, available in 2003, similar to my comments about hepatitis C, much of those advancements really just weren't applied. Everyone really got comfortable with the fact that HIV in many people was going to be a progressive illness, and there's complexities with diagnosis and therapy. And I think really got lulled into what I call status quo, just accepting HIV as an infection that's going to occur in some individuals, and we'll just do the best we can in the clinical scenario. There wasn't that view that is really kind of the under theme of what I want some of the younger people listening to take away from this. The idea that it was actually possible that you could actually bring an end to the HIV epidemic in the United States and then in countries around the world. People didn't see that as practical. I know that one of the reasons I accepted the job to be CDC director is that I believed that it was possible to bring an end to the AIDS epidemic in the United States and beyond. And we had all the scientific tools, just like hepatitis C, we had all the scientific tools. We just weren't using them effectively because the average individual, and it went all the way up to the top, didn't believe that bringing an end to the AIDS epidemic was possible. And we're going to talk a little bit about that. One of the things that we got lulled into is as therapy became available, we gradually saw decreases in the number of US cases from between 45 and 50,000, but then it really began to stabilize. So we had really somewhere between 35 and 40,000 new people every year being diagnosed with HIV infection. And it just, there was no more progress that was really made in the decade before I became CDC director. And everyone just became accepting that we're going to have, in the order of 40,000 cases a year. And that's sort of the best we can do. I had written, and this is when I was much younger, this is hard to believe because it has been 35, 36 years since I focused my career on HIV. But in 1986, I was asked to do a piece for US Medicine. And I obviously started with the quote that I already started this talk with about preventive medicine and what it dreamed. And I briefly presented in about 3 or 4 pages, encourage some of you that want to read it, that there was really a straightforward public health approach that we could take to HIV. And the medicine should embrace it. It's hard for some of the young people to go back there, and they may have different alibis for why this happened. But in those early years, I was confronted aggressively by the FDA and others that I was not to use the HIV diagnostic test for people; it was only meant for blood. And of course, as a young doctor at Walter Reed, I argued that I was going to use whatever test I could to accurately diagnose the men and women that I was trying to take care of. And because we applied HIV diagnostics in a way driven by clinical presentation, so if you presented with thrombocytopenia or chronic lymphadenopathy or oral candidiasis, we didn't decide whether we were going to test you for HIV based on whether you're a gay man or an IV drug addict or a Haitian or hemophiliac. We did it based on the clinical presentation. And lo and behold, this is one of the reasons back in the early 80s, a third of the patients that I served at Walter Reed in 1986 were actually women. And most of the other groups saw this strictly as a gay man's disease at the time. I just say that because in 1986, it was clear what the public health approach should have been to HIV infection. But I can tell you that it wasn't embraced. You had laws, some of the more sad laws that you had. They made it a felony in California for a pediatrician to be told by the obstetrician that the mother had AIDS. That was up to the mother to let the pediatrician know if she wanted that pediatrician to know. Otherwise, he should just take care of the child based on whatever knowledge he had. I was in Supreme Court cases where in New York, where a gentleman wanted his conjugal rights while he was in prison, but he had HIV infection, but there was a confidential requirement that couldn't be released. And he insisted on his conjugal rights and that his spouse was not told of his status. It's hard to believe, but that went to the Supreme Court of New York, where the health commissioner in the state and the health community all testified under oath that the HIV was not a sexually transmitted disease, and therefore he should not be denied his conjugal rights. And yet the justice group have voted 5-4 that despite that the health department didn't see HIV as a sexually transmitted disease, they saw it as such. And the rules were in New York, if you had a sexually transmitted disease, you had to inform your partner to get your conjugal rights. So this shows you how misguided much of the 1980s were. And I say that only because we're dealing with another pandemic now, and part of the challenges is how do you take high quality public health medicine and science and integrate it into effective public policy. I want people to realize it wasn't that different. It took a long time for public health policy to get aligned with the principles of medicine, and CDC didn't recommend routine testing for HIV, for individuals in clinical settings until 1996, actually 10 years, almost to the day after I wrote this original piece, suggesting that early diagnosis was going to be key to our ability to have an effective public health response to HIV. One of the things when I became CDC director was I did believe very strongly that bringing an end to the HIV epidemic was possible. I will tell you that everybody thought it was pie in the sky and that I was not in the real world in suggesting that was possible, that we were just learning to come to reality that 40,000 new individuals would be infected every year. It was interesting that HIV was fairly concentrated and largely in younger people between, say, the age of 18 and 35. If you looked at new infections in the different groups, and in the southern part of the United States. So it wasn't like it was totally diffuse. But when you asked to get maps of HIV infection, and which I did as CDC director, the epidemiologist would give me maps of cases per 100,000. And the map would look like HIV was all through the United States. And it looked pretty complicated to be able to see how we could focus. Being clinically trained, I asked the CDC to redo the map, and I didn't want them to do it per 100,000. I just asked them to plot every single infection so I could see where the infections were. And it was one of those moments that actually ended up changing policy. And this is the map. This represents 50% of all the infections in the United States. So when people saw this map, they said, wait a minute, it's not like all over the United States. It's like it's concentrated. And within those areas, it was concentrated in the 20 to 35-year-olds, and it was concentrated in certain groups, particularly African-American Hispanics, that you really did have a defined population that if you wanted to put intensive medical public health resources, it was really conceivable that you could begin to make meaningful impact. Now, fortunate for me, a couple cities started on this at the same time and started to have data showing that they could impact the amount of HIV infection by a series of pretty straightforward steps: diagnosing the disease, encouraging diagnosis, people that got diagnosis basically treating them the day you diagnosed them, and then aggressively beginning to institute pre-exposure prophylaxis prep in the population that remained at risk. You could reduce it. And the District of Columbia actually showed a 50% reduction in a year, and New York showed a 25% reduction. So people who said it wasn't feasible, there actually was several health districts that actually showed. And then again, I'm not a critic of DC, but it's not the most overwhelming, overstaffed health department in the nation. And they were able to put a program together that has substantial impact. And so it really was this map that, when presented to the leadership all the way up to the president, that we had the tools we needed to do to bring it in. We've had those tools since 2003, and we can diagnose the disease. We just have to get people diagnosed. And it's sad when you find out that, maybe 30% of the people diagnosed were probably infected for more than 5 years. 25% had advanced AIDS at the time of diagnosis. It's hard to believe that that's the case in 2018, but that's reality, because it wasn't being converted. This science was not being converted to impact the human condition. You could get people treated and get their viral load undetectable. But really, when we started this, we had many parts of the nation whose viral suppression rates were less than 70%. And those of us that worked in Africa, we were able to get our viral suppression rates in Africa over 85 to 90%. And yet in America, we had many groups whose viral suppression rates were less than 70%. So put an emphasis on getting viral suppression rates up, which largely meant putting an emphasis on the care delivery structure that you used, not something that the United States medical system seems to invest heavily in, but obviously fundamentally critical, which is to make sure not only that people have medicine, but they can be empowered to take it properly. And then the final was the fact that we knew that treatment was prevention. So the best way for me to prevent HIV transmission is to get everybody who's HIV infected into treatment and get them virally suppressed. And then those individuals that still have potential risk exposures, to allow them to now get on chemoprophylaxis, since we now knew that chemoprophylaxis worked in preventing HIV transmission. The problem is, again, it was not really readily applied. There are many physicians caring for patients that didn't necessarily take a strong history to try to define, was there a risk for HIV, and should these individuals be offered pre-exposure prophylaxis? But this just gives you a sense. And so one of the things that we were able to do was to make the case that beginning to bring an end to the HIV epidemic in America was in fact possible. It required investment, focused investment, to see the science applied into clinical medicine. And we were able, although people said it was not going to happen, we were able then to get the president to make a presidential initiative, which will put billions of dollars and more new funding to HIV that had happened in years, in a very targeted way, initially in these 50 counties, to begin to demonstrate that you can make an impact and reduce. And it's now in the area that you all are in. A number of the counties were in our area, as you can see. Actually, if we had cut the cutoff a little and gone to 60 rather than 50, we would have picked up 2 more counties in the Baltimore, Washington metropolitan area. But and then that program is ongoing, and it continues to make progressive progress towards the goal of reducing new infections in HIV by 2030 to basically less than 3,000 in the nation. So a little summary, we can come back and talk about it if people want, but science was enormously powerful in giving the tools to understand HIV. Science was enormously powerful, coupled with the private sector, to develop therapeutics, so that what was a largely progressive fatal disease, and then went into a time when it was treatable, but very complicated and only for a certain period of time. Death still was a not all too uncommon outcome, to now where basically people living with HIV infection really can't expect to live a natural lifetime, and in a therapeutic modality that is, very easy to accommodate uh, for effective treatment. And that we are, in truth, in the United States, on a path to the beginning of the end. And there's other countries that are moving, and they're moving quickly to begin to see it. And we are seeing the impact in other parts of the world. Clearly, we've seen a reversal in the mortality, and life expectancy has gone up in sub-Saharan Africa. When I started working in sub-Saharan Africa, less than 5,000 people had received access to antiretroviral therapy. I had a small program in Malawi that I was treating 93 people. And today we're over 27 million people that are getting regular therapy because of the ability to get therapeutic advancements into a highly effective globally available treatment. I'm going to contrast that a little. Initially, when I had this opportunity, I was going to focus also on the drug use disorder epidemic, which is an epidemic I spend an enormous amount of energy as CDC director. And it's a complicated epidemic in many different ways. It's also an epidemic that we all, and again, this is not to be critical of us in the medical community, but the medical community obviously contributed historically to the opioid epidemic. I know as a young doctor, I didn't know if you prescribed, order of oxycodone to someone that had a wisdom tooth extraction, that 3 to 5% of those individuals would go on to be addicted for life. I know I didn't prescribe a lot of narcotics, but I think if I had known that, I would probably hardly prescribe any, because I do think that opioid addiction is a very serious thing. People know I almost lost one of my 6 children, who's now in his 6th year of recovery and doing well. But opiate addiction, and who knows, that opiate addiction may have started with the wisdom tooth extraction where he got his 30 days worth of OxyContin. But in the interest of time, I decided I wasn't going to get knee deep into drug use disorder, but it's a big issue. It's a big epidemic. There's a critical role that the public health medical community plays in it, which I think in general we've not engaged as fully as we should. We argue, and I argued as CDC director, that stigma is the enemy of public health. Obviously, having witnessed it in AIDS, but I witnessed it in drug use disorder at a level that I've never seen before. And that stigma comes in the language that we use. You're an addict. And that stigma even comes in the way we approach it. A lot of times we push off treatment for addiction. We push that off. And I'm not saying psychiatry doesn't do a great job. I think it's a good job, but I think addiction should be incorporated into clinical medicine as much as we can. And I know even when I've watched a lot of young people try to get treatment, that one of the things that basically shut them off right away was you need to go see a psychiatrist. And so that's another area. But COVID-19, the other premonition I had when I was asked to be CDC director, having 14 grandchildren, was I did feel that we were in for the potential of a great pandemic. Now, obviously, I felt it was going to be bird flu because that's what happens. That's the evolutionary epidemic, and we'll come back to that at the end. Unfortunately, I do believe that we're in line for the great pandemic. Right now, we're in what I call the lesser pandemic, which is COVID-19. I do think we're in line for the great pandemic, which will be a bird flu pandemic, which we can talk about when we have discussion. So the idea of being CDC director and as an infectious disease virologist at a time when I thought there may be great risk for a pandemic was something I felt, was worth the personal professional risk of going to Atlanta and trying to see and make sure our nation was prepared. I think one of the things that's clear to anyone that's looked at this critically is our nation's not prepared, that we do not invest in public health the way we should. It's an afterthought. I do believe that the risk to the American way of life is much greater likelihood to be impacted by pandemics and epidemics than it is by some military action. And then I'll continue to be a big advocate to get our nation to have a proportional investment in public health. Proportional. I've argued at Congress many times that I thought they should at least put 5 aircraft carriers on the table, and let's start putting some real money into public health because we haven't invested in public health. And the truth is, it's not done at the federal level. It's not done at the state level, and it's a big failing that we have in terms of our ability. But on New Year's Eve, I got a phone call from my team that has a CDC in Beijing next to the Chinese CDC. And the Chinese had seen 27 cases of a new non-specified pneumonia, which was not flu, and it was not SARS, and it was not MERS, and it was not adenovirus. So it was an unspecified pneumonia. And they were beginning to evaluate to try to understand what the etiology of this was. And one of the lessons that hadn't been learned from HIV, if you go back and read what happened then, is I'm talking to my counterpart, who was the head of CDC and at the China CDC, all 27 patients had exposure at the wet market in Wuhan. And he was confident that there was no human-to-human transmission. But he was kind enough to share with me the histories of the 27 patients. And I noticed right away that 3 of them were clusters: husband, wife, child, 2 children, and a mother. So those clusters, I said, George, maybe there is human-to-human transmission. He felt pretty confident there wasn't. They didn't see secondary spread in the hospital. I'm pretty confident that there was no human-to-human transmission. And he was totally confident that all the cases were linked to the wet market. So I asked George just what the case definition was for these new cases. And he said, unspecified pneumonia, not flu, from people who came from the wet market. And I said, George, by definition, everybody's from the wet market. And it's the same thing we did in HIV when people said you test people for HIV that are either hemophiliacs, Haitians, or homosexuals, or IV drug users. And of course, I did differently at Walter Reed. I tested everybody with leukopenia, with thrombocytopenia, with chronic lymphadenopathy, and I found out there were actually a lot of women had HIV infection. I advised him to go look for people without the wet market exposure, and 3 days later, he called me and he said he had found several hundred people with unspecified pneumonia that had nothing to do with the wet market. And the wet market really was never reality. I can't say why it was promoted. I have my own suspicions, but it was never reality. And by the 10th of January, it was clear that there was a widely diffused respiratory pandemic going through Wuhan. And many of you probably saw the pictures and the rapid setting up 1,000, 2,000, 3,000 bed hospitals and quarantining literally 11 million people. That didn't happen because of the wet market. It happened because there was a highly transmissible new respiratory pathogen that originally had very high mortality in Wuhan, probably over 8 to 10%. I think a lot of that was not mortality due to the virus, but mortality due to a health system that didn't function. We saw the same thing in New York in March. Many people may not know, but the pandemic in America, in New York City in March, had a mortality rate of 8%. It wasn't really the virus that was killing 8%. It was the health system that was getting overwhelmed. So people were missing a lot of the key observations that they would normally pick up on so that they wouldn't end up accelerating. The second comment I want to make is jumping to the wrong conclusions. So the first conclusion, to compare the difference between HIV that we talked about that took 3 years to figure out etiology, methods of transmission, develop diagnostic test. We understood the etiology of this new unspecified pneumonia that was first formally recognized December 31st, it was formally reported with molecular sequencing on January 10th. So basically in 11 days. It's pretty remarkable. Contrary to the news, the CDC test was never defective. We can talk more about that. The CDC developed an excellent test by January 17th that we have today, works perfect, and it worked then. It allowed the ability for us to diagnose this new infection. So it didn't take us 3 to 4 years like HIV. It literally took less than 2 weeks from the time that the molecular sequence was available. But the scientific community, and including ourselves, including CDC, including NIH, we immediately called this virus SARS-like. And I think many people don't realize what a big mistake that was. Because when you think about SARS, what do we know about SARS? SARS was the prototype coronavirus that came in about 2003. And it came initially from a bat into civet cats into humans. But one thing SARS never learned how to do is it never learned how to go human to human. Many of you might not have emphasized this, but if we sit here today, 18 years after SARS had came into the human species, we've had less than 10,000 cases. And in the year that the SARS epidemic started, 2003, 2004, there was less than 1,000 deaths. And what happened to SARS is it disappeared. It was one of the reasons the private sector didn't get very excited about COVID-19, because they got excited about SARS and developed some investments both in diagnostics and therapy and vaccines, and the pandemic disappeared. Same thing happened for MERS in 2012, where it went from a bat to a camel. And it also has, caused mortality in less than 1,000 people. And it also has more or less disappeared. Now, one thing about SARS and MERS, or being SARS-like, SARS doesn't, when it infects a human being, it makes the human being sick. And about 7 to 9%, 7 to 10% die. Or MERS, when it infects a human being, it makes them sick. And over 30, 35% die. This virus was not SARS-like. It was a whole different virus. It was not a virus that basically made you sick necessarily. It could infect you and you could get not sick at all and still be transmissible. So the public health response that was developed on that decision that this was SARS-like was that we would identify symptomatic individuals, and then we test them and isolate and contact trace around them, evaluating context for symptoms. And if they develop symptoms, then testing them. We started a program right away. I was very disappointed that the Chinese government didn't allow us in. I had a large team ready to go in the first week of January, and we're able to get permission from the Chinese government, despite the president directly asking the president of China to allow us in, because we wanted to learn very quickly whether there was asymptomatic transmission, was there human-to-human transmission. In the 14 cases that I diagnosed in the United States in between January 20th or January 19th and the middle of February, we had over 800 contacts that we evaluated. And only 2 of them were infected with COVID-19. So it gave people the view that this was not that infectious. It was SARS-like. But the problem was the public health response was a SARS response. We didn't test 800 people. We tested the 2 people that were symptomatic. And I suspect if we could get a do-over, we would have found that a lot of those contacts were infected, because the dominant transmission for this virus is not symptomatic transmission, it's asymptomatic transmission. And that focus, we didn't get to that focus until the end of February. And why is that important? Because it totally changes the public health response. The idea of containment was looking for symptomatic individuals and containing around them. The problem is the majority of people with COVID weren't symptomatic. And so what it really meant is a different strategy should have been launched, which is aggressive serial testing to identify the asymptomatic individual and pull them out of the transmission pool. And that the public health response that was driven by the fact that this was anointed as a SARS-like pathogen then triggered a whole series of public health responses that in retrospect were in the wrong direction. I'm proud of as part of the the Operation Warp Speed project to develop vaccines, but the truth is we should have had an Operation Warp Speed diagnostic project in the end of January to provide billions of tests, because the defense that we had from a public health point of view, and the one that we should be moving to now, although we're still going way too slow, is basically regular repetitive testing to identify the asymptomatic infected individual and pull them out of the transmission pool. Once we knew asymptomatic spread was really going to be driving this, we changed focus to try to, at least we knew we weren't going to contain this pathogen anymore, but could we slow it down? And many of you remember the attempts that we had, the 15 days to slow the spread and then the 45 days to slow the spread, with the idea that that would give more time to develop countermeasures, both monoclonal antibodies, potentially antivirals, and most importantly, vaccines. Warp Speed was a very special experience because it's another example of people who didn't see this as possible. And I'm not going to name names, but there were many experts that made it very clear to the president that we wouldn't have a vaccine sooner than 3 years. The mission that was assigned was to have a vaccine by January 2021. And in 7 months, basically 4 vaccines were successfully developed. 3 of which are approved for use in the United States. And it was that was really done largely by the private sector, and it was done by the federal government providing resources and the Defense Production Act. But it was really done because people said it could be done, rather than people saying it couldn't be done. And we can come back and there's questions on that. But I think it's remarkable that those vaccines were developed. What many people may not know is one of the reasons it was so successful is after phase 2 trial, going into the phase 3, the United States government offered the manufacturer and purchased 100 million doses sight unseen, so that the manufacturers like Pfizer, Moderna, J&J had the resources to build the manufacturing plants that if their phase 3 trial worked, they would already have product, as opposed to the regular process where we would wait till the phase 3 was over, the FDA would review it, they would say it's acceptable, and then the company would begin to build manufacturing capabilities. This was all done basically in June, July, while the phase 3 trial was just being initiated. Other comment I wanted to make here about jumping to conclusions, you've probably heard a lot about it, where there are people who were pontificating that, all we had to do is to get to some level of immunity, herd immunity, and this thing would go away. I tried to argue as a virologist that that's not an operational for COVID-19, that the durability of protective immunity from natural infection is non-durable. And that we should anticipate that the durability that was induced by the vaccines that are highly effective, but they're going to have a durability problem. And it's just developing a durable response to this glycoprotein is something that the human immune system has trouble doing it. Some of it may be just by design, and if there's questions, we can go into it. This virus has some very unique issues in it that have raised concern about origin. But one of the things for sure this virus has is some of the key glycoprotein domains are masked. The immune system doesn't see them well. It's probably very similar actually in some degree with HIV, having a major problem of durability. If you can get an effective immune response, it's just not durable. Clearly with COVID-19, it's not durable. If we get naturally infected, we'll be protected for a period of time, probably less than 6 months, and then we're susceptible to reinfection. If we get vaccinated with an approved vaccine, Pfizer, Moderna, J&J looked like it was probably only protective for about 2 months, maybe 3. The mRNA vaccines are probably protective for more like 6. So it's important to realize that we're not really going to be into this one and done herd immunity. We're going to be into a cycle of maintaining immunity. And obviously some of the key scientific questions that are needed right now as to how to have the diagnostic tools for all of us to know what that really means, as opposed to the date that you got vaccinated. A lot on the origins of COVID are kind of important in here. I mean, this virus is is different. I don't personally believe, although people think it's controversial, but as a virologist, I don't believe this virus evolved naturally from bats into some animal we haven't found. I think there's a lot of evidence that this virus got into a laboratory, and there were laboratory experiments that were being done that enabled this virus to gain a greater tropism for humans. Probably one of the most important signature sequences, and it's declassified now, so I can comment on it. But very early on, we learned that this virus had a furin cleavage site, which the other bat coronaviruses do not have. And the reason it's important is that cleavage site changes the orientation of the binding domain of the virus for the ACE2 receptor. So if we do it the way it naturally is without the cleavage site, this virus doesn't bind human H2 receptors. If you have the cleavage site in place, now it binds human cells, but it doesn't infect, and it does no longer infects bat cells. So that just is something that I think is consistent with the papers that have been published by the laboratory, where they were intentionally adapting bat coronaviruses to humanized mice to bind the H2 receptor. That's all published material, so it's not speculation. Now, why is that important? Because this virus got a head start. It knew how to infect humans from the beginning. As I mentioned to you, SARS, we've had less than 10,000 cases in 18 years. MERS, we've had less than 10,000 cases in 8 years. Those viruses still to this day haven't learned how to infect humans. And so you ask me, well, where are all the variants to SARS? There's not a lot of variants to SARS. Where are all the variants to MERS? There's not a lot of variants, because those viruses don't replicate well in humans. But unfortunately, this virus does. That's why the variants are the rule. And we predicted that after Delta, there would be a new variant, but that new variant would have some very special characteristics. They would have a replication advantage, not in the naive individual, but a replication advantage in the partially or fully immunized individual. And I think that's what you're going to see with the current variants. The future, we're going to continue to see variants. The future, there's going to be an enormous opportunity now to go through what we saw with HIV and therapeutic progress. I think that's the name of the game over the next 5 years, to really get more and more and more effective antiviral therapeutics. And finally, adjusting ourselves to the new normal. This virus is going to be with the human species, for the duration. And we're going to have to learn how to confront it. It's going to change and try to outmaneuver our countermeasures. But I think it's a poise for the power of science to begin to bring this pathogen back under control, largely by trying to get improved vaccines that have greater durability, which is a real strong, important scientific question, and of course, developing antiviral therapeutics. So I think the last one I wanted to show real quickly, this is just to remind you, our mitigation steps really probably did work. This is a curve of looking at COVID, and the scales are different. On the one side, COVID in the blue, and just shows you the peaks of the COVID pandemic that we had. But what I want you to look at is the red line. The red line was what happened to flu during the first year of our mitigation. We didn't see flu. So the mitigation steps we used were highly effective in blocking flu transmission. You can see they weren't as effective in COVID, but it shows you that flu is really susceptible to masking, washing our hands, and social distancing. On the other side of the slide is looking at flu on another scale in the other couple of years. But you can see the first year of COVID, we didn't see flu, and we didn't see RSV. Both of these viruses were totally shut down by the mitigation steps that we practiced initially. Unfortunately, they're coming back now. As you can see in black is showing you RSV coming back now. But I just wanted to show that because I thought it was useful. And then finally, I just conclude with something I've always stressed, is you never underestimate the possible. I'm always tried to use this when I tried to argue that we could bring an end to HIV in America. Remarkable, John Kennedy, when he said that he was going to get to the moon at the end of the decade, remarkable that basically Neil Armstrong got there before the decade was over. I would argue largely that was driven by someone having the leadership vision to say it's possible and then leading the nation to act. The same thing, I will argue, is true for bringing an end to the AIDS epidemic, which we're on track to do in the United States. And the same thing I would argue is to begin to develop the countermeasures that we need to put the coronavirus, if you will, in its place. We'll never get it back in the box. It's out, it's going to be with us for the duration, but we can make it so its ability to impact health. The virus will continue to evolve to be more infectious, like we're seeing with the Omicron variant right now, but at the same time, these viruses are going to be evolving to be less pathogenic. That's what viruses do. They don't want to get more pathogenic because they have to replicate in the host. So I want to thank you for the opportunity. I hope my comments were of some value, and if there's time for a couple questions, I'm happy to take them.
Thank you, Dr. Redfield. Dr. Davis is also on the call. So, if you'd like to say a couple words, you can unmute. We'll also be taking some questions in the Q&A. We have a couple minutes.
So Bob, Dr. Redfield, thank you so much on behalf of the Department of Medicine and the ICTR. It's wonderful to see you back, and we thoroughly enjoyed your presentation, your thoughtfulness, your provocative thoughts and new information. And so I want to hand it back to the chief residents, and any questions, which I'm sure there are, we'll be able to discuss further.
All right, thank you, Dr. Davis. So we have a question here for you, Dr. Redfield. Do you think there are strategies we can apply for public health preparedness from the military that would benefit the civilian population?
Well, I think the preparedness is critical. I do think our nation is non-prepared. And, I've had multiple congressional testimonies to try to get Congress to realize that how unprepared we are. And one of the challenges is everyone, I think, was prepared to actually make huge investments in public health. And then, of course, when the vaccines came, everyone kind of thinks, okay, this is done. We don't have to worry about it again. We're at high risk now for two reasons. We're at high risk from the evolution of pathogens, particularly bird flu, which I think will be much more difficult for us. Unlike COVID, it's not going to spare mortality in younger people. It's going to be a fairly fatal pathogen for all of us. And it's going to spread basically as fast as COVID did. For those of you that don't have an appreciation, it's been less than 2 weeks from the new variant was first found in South Africa, and now it's over 70% of all of the isolates in South Africa are that variant within 2 weeks. And of course, that variant now is around the world. And it will probably become the dominant variant everywhere, including the United States, within the next 12 to 16 weeks. I do think the military has a lot to offer. Obviously, I spent over 20-something years there. But I do think there's so much to getting prepared that our nation has not demonstrated the interest in investing in. When I was trying to start our contact tracing program for COVID, I had some states that had less than a handful of contact tracers. Our state's public health labs have no redundancy in technology. The reason we developed the original test using a low throughput Thermo Fisher system that was similar to what we use for flu is that's all that the public health labs had. There was only one public health lab in New York that had a high throughput system. Nobody has redundancy in laboratory technicians. Nobody had redundancy in the public health. Our data system, when we had the measles outbreak that I met when I first became CDC director, I actually had health departments sending us data on their cases by fax machine. So there's a huge requirement to invest in public health infrastructure in this nation. And unfortunately, I don't see the political will to get it done. I mean, I'm going to still try to articulate it and argue for it, but I don't see the political will.
It looks like we have another question here. As you reflect on your experience as CDC director, what was your biggest challenge in dealing with COVID as you observed the long-term impact of this virus?
I think, a fundamental disagreement. I firmly believed that we should have had a national response. And obviously, as CDC director, I would have liked to lead that national response. It was decided that it would be a federal assisted but state-led response. And in some states, California, Maryland, a lot of these states, they have independent health departments. So it actually was 37 different responses, and they didn't agree on a lot of the principles. So I think the biggest frustration I had was the inability to really launch a national public health response to this pathogen. I argued very strongly as CDC director against closing schools. Most of the governors didn't listen to me and closed schools immediately. I was very gratified, and I got a nice award. I was able to keep the parochial schools open in Baltimore. They listened to me, and they were able to keep them open. And people didn't realize I argued that the public health harm done to these kids K through 12 for closing schools was huge. Many of them got their nutrition that way. Many of them, 7 million got in mental health services through the schools. Basically, we saw drug use and suicide go up when these kids, a lot of them are basically off the plane now in terms of their learning. They're not going to get back on the track very quickly. It was a huge harm that was done, and that was facilitated by the lack of having a national response. I mean, it was a very active decision that was made by the administration that this would be a state-led response. I just think that was the wrong decision.
All right, I think we've don't have time for any more questions, but well, thank you again, Dr. Redfield, for your time
Thank
and
you.
for your presentation.
Thank you very much. God bless you all.
Thank you, Bob.
Thanks, Steve.